Well, hello again, everybody. Welcome back to our ongoing discussion of the material for BSC1010C. This time we are taking a look at cells, and we're taking a look first at a big picture view of cells, and then we're going to narrow down our view to eukaryotic cells and talk at length about the structural features of eukaryotic cells and what they do, their functions. So this is going to be an overview of a typical eukaryotic cell, more or less. We do discuss very briefly the differences between prokaryotic and eukaryotic cells, probably something that you're already familiar with, but we will touch on that. We'll talk about some of the reasons why eukaryotic cells have the internal structure that they have versus that of a prokaryotic cell right at the very beginning. But most of our focus is going to be on those standard features that you would expect to find in any typical eukaryotic cell, irrespective of where you were looking. And then we'll wrap it up by summarizing everything that we've covered throughout the content of this presentation. Hopefully this is not going to be as long as the last one. Yeah, that was quite a long one, and I can tell you that putting that presentation together was probably a little bit more painful for me than perhaps you watching it. I don't know. But certainly I don't want to do presentations of that length ever again if I can avoid it. So we'll see how we get on with this one. If it's starting to get too long, I may break it and split it into two parts. But we'll see how we go. So that being said, let's start at the beginning by talking about cells in general. So one of the overarching themes that we discovered back in chapter 1 is the idea that cells represent the smallest unit of life. If you want to know what is the smallest unit that you can find in living organisms that have or that has all the characteristics that we assign to a living organism, then the cell is it. That's the smallest unit that has all the characteristics that we assign to living things. And so if you cast your mind back to what we talked about in chapter 1, you'll recall that there were a list of things that we recognize all living things do. And those are the features not only of all living things, but of all cells as well. And this is encapsulated in a theory that goes back to the mid-1800s called the cell theory of life. And it states that all organisms are composed of cells, nothing mysterious there. And most people recognize that. But there is a second part to that theory. And it says that all existing cells arise from pre-existing cells. So by implication there, we are saying that cells are capable of reproducing themselves, irrespective of whether they are part of a single organism, a unicellular organism, or whether they are part of a multicellular organism. Cells have the ability to reproduce themselves because that would be the only way for existing cells to be derived from pre-existing cells. There must be a way for cells to duplicate themselves. And we will address that issue when we get to chapters 12 and 13, because we'll talk about the two different types of cell division that eukaryotic cells are involved in. So more to come at a future date about that second part of the cell theory. But today we just want to talk about cells in general with an emphasis on eukaryotic cells. So even though we do recognize that there are two very distinct types of cells, prokaryotic cells, which we find constitute bacteria and archaea. They are the two domains that feature prokaryotic cells. And eukaryotic cells, the cells that are the constituent cells of organisms classified in domain eukarya. But there are common features that are found in all cells, irrespective of whether they are prokaryotic or eukaryotic. So the first thing is every cell has a plasma membrane or a cell membrane. Every cell's got to have one. and it demarcates the inside of the cell from the outside of the cell. It separates the cell contents from the outside of the cell. And as we discovered in the previous chapter, the molecules that constitute the framework of the cell membrane or plasma membrane, the phospholipids, are a perfect molecule to do this because they are amphipathic. They have both a hydrophilic and hydrophobic domain or region. And the hydrophobic domains cluster in the center of the membrane, creating a hydrophobic barrier between the water of the environment outside of the cell and the water of the environment inside of the cell. It's a perfect arrangement. But because there's water on both sides of the membrane, the hydrophilic regions of the phospholipids point outwards, out to the water around the outside of the cell and out to the water that constitutes the inside of the cell or the material inside the cell. That material inside the cell is referred to as cytoplasm or cytosol. So let me just write that in here. Let's fire up the pen and get it working. And so this is also known as cytosol, and we could also say here that the plasma membrane is also known as the cell membrane. We might as well put that in there as well. And the terms are interchangeable. So you can say plasma membrane or cell membrane, you can say cytoplasm or cytosol. And it's really hard to explain what cytoplasm or cytosol is like. It is a water-based solution, but it's not liquid as we would expect water to be. The best explanation that I can give, and you're going to have to go with me on this one, is when you make Jello gelatin, you know that you heat the water up and you dissolve the crystals in there, and it's very liquid at that point. You know eventually it's going to set into the gelatin, and that's going to be a semi-rigid solid. But somewhere in the middle, between it being truly liquid and it reaching its final consistency, it's going to be neither liquid nor the solid form. It's going to be somewhere in between. And that's really what cytoplasm is like. It's neither liquid nor solid. We say it's semi-fluid. And it's really confusing to really describe what that truly means. But this intermediate stage of Jello is the best that I can come up with. So if that resonates with you, great. If you can come up with another idea that seems kind of the same, that works for you, by all means, go with that. But just remember that the cytoplasm is not liquid like water, nor is it solid. It's somewhere in between. It's this semi-fluid arrangement. Embedded in all of that is going to be the organelles that we are going to find primarily in eukaryotic organisms, but there is a type of organelle that is also found in prokaryotic organisms. And there's some disagreement, I guess, as to whether these constitute organelles or not. And these are the ribosomes. We'll talk a little bit more about those because it appears that a number of people are now saying that if you want to describe an organelle, the first thing that it must have is a membrane around it. And ribosomes do not have membranes around them. So there seems to be a trend to redefine what is truly meant by an organelle. I'm not sure how much traction that has in the cell biology field and in the larger field of biology in general, but I know that the textbook has tended to trend in that direction. Being somewhat old school myself, I still consider ribosomes to be organelles, and therefore they are also considered to be common amongst all cells because they are the machines that assemble polypeptides and proteins. And all cells make polypeptides and proteins, which means all cells need ribosomes, irrespective of whether they are rightly described as organelles or whether they're described in some other fashion. Personally, I think they still should be accepted as organelles, but that's my position. And then another common feature that all cells have, and this is in fact a common characteristic of all life, as we discovered back in chapter 1, is the presence of DNA as the molecule of inheritance, the genetic material. And so every cell, every organism uses DNA as its genetic material. Now, if you're going, but didn't we say that there are viruses that have RNA as their genetic material? Isn't that something that we mentioned in passing back in chapter 1? And the answer to that is absolutely, that's true. There are RNA viruses. But viruses, are not considered to be organisms. They're something else. And in fact, that's one of the struggles in biology, to try and figure out where do you actually place viruses in a taxonomic system like the one that we discovered in chapter 1. You would notice, if you did some investigation, that viruses generally are not included. They do have their own taxonomy, but it has been up until recently considered to be completely independent of the taxonomy of organisms. But there are attempts now to try and find a way to incorporate viruses into the general taxonomy of life to make sense of them. But I'm waffling, I'm rambling a little bit. I tend to get a little bit diverted from the topic at hand, as you may have noticed, but I'm trying to make some connections for you. So if you're ever wondering why I tend to go off on tangents at times, and it may seem to be not really connected to what we're talking about, it's because I'm trying to make connections between what we have in front of us in terms of the course material that you are required to master, and connections to either your future professional life or just your life in general. So somewhere along the way, it helps to have those connections because it makes the information more valid, more usable, and therefore more readily learnable. So if you're wondering, that's my story, and I'm sticking by it. So these are considered to be the common characteristics of all life forms. I'm just going to add a note in here. Ribosomes are also universal. So again, irrespective of whether you consider ribosomes to be organelles or something else, they are universal. So we'll put these in because that's a common characteristic of all cells, the presence of ribosomes. So here are the features of cells, and not surprisingly, they are the same features that we saw in as common themes as far as life is concerned. So no mysteries here. These are the same characteristics. You should be familiar with all of these. So we will just move on from here and continue talking about cells in general. So we know that there are these two different types of cells, prokaryotic and eukaryotic, and the names actually tell you something about the cells. So so another prefix to replace pro with the pre. And most people know what pre means before. The term karyotic comes from a word karyon, and that's a Greek word for nucleus. So if we look at the word, prokaryotic means pre or before the nucleus. And this describes a prokaryotic cell pretty accurately. This is a cell that lacks a nucleus. Doesn't mean that the DNA is found randomly anywhere inside the cytoplasm. It actually is located in a specific region called the nucleoid. This is an area of the cytoplasm that seems to be free of other features other than the DNA. There are no ribosomes in here. There are no other obvious features that we can detect in the nucleoid region other than the presence of the DNA. So this seems to be a section of the cytoplasm that is set aside for the DNA, but it's not surrounded by a membrane. So it's not separated from the rest of the cytoplasm. Then if we look at the cell as a cell, we know that it's going to have a plasma membrane or a cell membrane. And then because of the environment that prokaryotic cells live in, environments that are largely water, as you'll learn when we do the next chapter, chapter 7. Under those circumstances, water is bound to move into the cell. And if there wasn't a way of protecting the cell from this constant influx of water, it would swell up and burst. So prokaryotic cells also have an outer layer referred to as the cell wall. It's semi-rigid, and it limits how far the cell can swell. And because you limit how far the cell can swell, it also prevents water from coming in. When we look at plant cells, we're going to see cell walls as well. Different constituent molecules, but basically doing the same thing. And a really good way of describing a plant cell is a cell inside a wooden box, because the cell wall is made out of cellulose. And as hopefully you recall from chapter 5, cellulose is the major constituent of wood. In fact, the cell walls of dead plant cells are the wood. And so really a plant cell is a cell inside a wooden box. So if you were to put a balloon inside a box, say a rigid box, and try to blow air into it, you could blow the balloon up a certain amount, but eventually the wall around the outside, the box around the outside, would prevent the balloon from swelling any further. And it wouldn't matter how hard you tried to pump air in, you couldn't get any more air in because the box resisted it. That's the purpose of the cell wall. And having a cell wall is a fairly common thing. In fact, there's only one group of cells that routinely do not have a cell wall around the outside, and that's animal cells. And there's a story involved in that we'll get into in more detail in chapter 7, because it's something that we cover in detail in chapter 7. But prokaryotic cells, whether they be bacteria or whether they be archaea, do have cell walls, different constituent molecules, but they do have cell walls nonetheless to prevent the cells from swelling up and bursting in the environments that they live in, which is largely water. Then we do know that a large number of prokaryotic cells, upwards of 50% of them, are also motile. That is, that they can move around. And in order to do this, they have flagella. Now, we're going to see flagella as a constituent of eukaryotic cells, or some eukaryotic cells as well, but they are totally different. So flagella here in bacteria are very, very different from the flagella that we see in eukaryotic cells. So they can't be considered to be, well, they're functionally equivalent, but they can't be considered to be structurally equivalent. They are very different. Then there are other features known as pili that extend out from the bacterial cell, and these are often used to grip onto surfaces. So a lot of prokaryotic cells like to anchor themselves to surfaces, and pili are one way of doing this. Other prokaryotic cells, bacteria and archaea together, have a sticky coating around the outside called a capsule, and that allows them to stick to surfaces as well. A lot of bacteria like to stick to surfaces. So you know that if you fail to brush your teeth for a short period of time, you start to feel deposits building up on the surface of your teeth. We refer to that as plaque. That starts by bacteria sticking to the surface of your teeth, and then saliva and food particles start to stick along with and start to build up, and the plaque builds up, and it's referred to as a biofilm. But it begins with some of the bacteria in your mouth, and there are always bacteria in your mouth, starting to stick to your teeth because you failed to brush. And that means you give them the opportunity to stick. And so it's the pili and the capsule, this sticky coating, that allows them to stick. So these are the common features that you're going to find in most prokaryotic cells. Again, whether they be bacterial cells or archaeal cells, they share these as common features. So here you can see a typical bacterial cell. You can see the pili, they're also known as fimbriae, sticking around the outside. Pili is actually an old term. We typically save the term pilus, which is the singular of pili, for a special feature that is a quote unquote sex organ. It's a connection that bacteria make with other bacteria to allow them to exchange genetic material directly. And it's referred to as a sex pilus. And so in the bacterial world, there is a tendency to reserve the term pilus for those and use this new term fimbriae for these extensions. So we might as well just use the term fimbriae. And these allow the cells to stick. Then you've got the sticky capsule around the outside. Inside that, you've got the cell wall, and then inside that, you've got the plasma membrane. Then you have the cytoplasm. Embedded in the cytoplasm is the DNA, along with ribosomes. And you can see the ribosomes as little balls here. And remember, again, ribosomes are considered to be universal to all cells because all cells need to make proteins, and that's what ribosomes do. And then we have the flagella, and you don't see this really easily on this slide, but if you take a look at the picture in the book, or if you can magnify this slide on your computer, if you're looking at the slides that you can download, then you can see a little structure at the base of the flagellum. This is actually a little electric motor, and it really is a little electric motor made out of proteins. What flows through it is electric charge in the form of hydrogen ions. And that allows it to spin around and around. And because it's spinning around and around, the flagella spins round and round and acts like a boat propeller. And it drives the bacteria through the water. And about 50% of all bacteria have these flagella with their little motors on the end of the flagella. There might be one flagellum, or there might be multiple flagella, but they all work together to drive the bacteria through their environment. And it's been described as kind of like us swimming through molasses for bacteria to swim through an aqueous environment. So you can imagine the sort of forces being generated by these little electric motors and the attached flagella. It's quite amazing to think about when you start thinking about that sort of thing. And they've moved through their environment in what is called a random walk. They'll swim in a straight line for a little while, then tumble, and then swim off in another direction, tumble and swim off in another direction, so long as they're not finding anything that's of value to them. As soon as they encounter something that's of value to them, or conversely, something that they'd rather get away from, they swim off in a straight line, either into the stuff that they like or away from the stuff that they don't like, until they notice that or detect that the concentration is decreasing. And then they return to the tumble, swim, tumble, swim, tumble, swim, which is called a random walk. And so that's how bacteria move through the environment. And archaea do pretty much the same thing as well. In terms of behavior, there's very little difference between bacteria and archaea. But there is structural difference and there's genetic difference. And there's biochemical difference. So there are a number of distinguishing features that really do separate bacteria from archaea, and that's why we recognize them as two different types of prokaryotic cells, something that is only relatively recent in the history of biology, because the first archaeal cell was not recognized until 1973. So the idea of there being two different types of prokaryotic cells is a relatively recent idea. And once it was recognized that there were two different types of prokaryotic cells, people started searching for more archaea, and we now recognize that the diversity of archaea is pretty much on par with the diversity of bacterial cells. They've both been around more or less for the same length of time, and therefore had an opportunity to start investigating and utilizing all sorts of bizarre environments. Where archaea seem to differ from bacteria is that the more extreme and the more bizarre types of environments seem to be the ones that the archaea are able to use. Whereas bacteria tend to be confined to more quote unquote usual environments, meaning not too hot, not too acidic or alkaline, not too toxic, not too depleted of oxygen, whereas the archaea seem to be able to tolerate and utilize more extreme things. So bacteria will live in salty conditions, but archaea will live in salty conditions that are 10 times saltier than seawater. Bacteria will tolerate heat up to around 90 degrees Celsius, but archaea can live in temperatures up to 140 degrees Celsius. These are the sorts of things that we've now discovered about archaea, which is the reason why archaea are sometimes known as extremophiles. But not all of them live in these sorts of extreme environments, so it would be incorrect to label all archaea as extremophiles. But nonetheless, there are these extremophile members of the archaea in there. So that's prokaryotic cells, and that's all we're really going to talk about as far as the structural features of prokaryotic cells at this point. I think I said eukaryotic cells just a minute ago, but I meant prokaryotic cells. So when we start looking at eukaryotic cells, the first thing that comes to mind, that becomes obvious, is that eukaryotic cells are significantly larger than prokaryotic cells. Now, that is a generalization. That does not necessarily mean that that is uniform, because the largest prokaryotic cell, I'll put this in here. So largest prokaryotic cell and it's a bacterium, is about 0.5 millimeters long. And at that size, it's just below the limit of size that our eyes could detect unaided. So these bacteria are just a little too small to be visible to the naked eye. We can see them under light microscopes, however, they're quite large. And being about 0.5 millimeter long, because they are the rod type of bacteria, otherwise known as bacilli, means that they're almost as big as as an average eukaryotic cell. In fact, they're bigger than some of the smaller eukaryotic cells. They're bigger than red blood cells, for instance. But this is unusual. This is not the norm. This is an outlier. Most prokaryotic cells are very, very small, and you struggle to see more than the basic outline of the shape in a light microscope. Whereas you can clearly see eukaryotic cells in light microscopes because they are large enough to be seen in light microscopes, and some of the really large eukaryotic cells you can actually see with the naked eye. You do not need any assistance, although a hand lens, a magnifying glass, helps a little bit, but you don't need a microscope to see them. The other thing that is very clear once you start looking inside a eukaryotic cell is that the inside of this very large cell is divided up into smaller compartments by membranes. Now, we used to define a eukaryotic cell by the name. So if you look at the name again, we've got the prefix EU or U followed by karyotic, and as we know, that refers to karyon or nucleus. There are a number of different definitions of U, as has been pointed out to me by a colleague who teaches Latin at the college. And he said that U can have a number of different definitions, but one definition of U is true. or real. And so a eukaryotic cell has a true nucleus or a real nucleus. And the reason why that was part of the definition was because under the typical light microscope, like the ones that we use in our labs on campus, and represents as a good light microscope, not an outstanding light microscope, but a good average light microscope. About the only feature that you can see inside a eukaryotic cell is the nucleus. It is relatively easy to see. But there are other organelles surrounded by membranes that we now know by using electron microscopes that allow us to magnify not a couple of thousands of times, but hundreds of thousands of times with a really good electron microscope. We can see that there are numerous membranes surrounding sections of the cells, in other words, dividing the cell up into compartments. And so the classic membrane-bound organelles that we identify are listed here: the nucleus, the endoplasmic reticulum, sometimes referred to as the ER, and you'll hear me refer to that consistently rather than talking about endoplasmic reticulum all the time. Golgi apparatus, mitochondria, lysosomes, and peroxisomes, and we will talk about all of these in detail. Then those that are not surrounded by membrane include the ribosomes, and so this is the reason why sometimes ribosomes are not considered to be organelles, because people are trying to redefine organelles as being bounded by a membrane. So ribosomes, microtubules, centrioles, flagella, and cytoskeleton. And it's these other ones of the non-membranous organelles that make me think that ribosomes need to be continued to be considered to be organelles, because these are all considered to be organelles too, but they don't have membranes. So I think there's a little bit of inconsistency at times when people say ribosomes are not organelles, but these other ones are. Because these don't have membranes, ribosomes don't have membranes. If these are organelles, then ribosomes are organelles. If ribosomes are not organelles, then these others shouldn't be considered to be organelles either. So I think there's a little bit of inconsistency going on here at times. But these are the major features that we recognize in eukaryotic cells. So what is the big deal about subdividing the inside of a eukaryotic cell into compartments? What's the advantage of doing this? Well, we see the advantage of doing this by considering the cell not as an irregular shape, or maybe even a circular shape or a spherical shape, but considering it as a cube. So this would be a model cell having a cubed shape. So if we look at a small cube, i.e. a bacterial cell, for instance, then when we look at the surface area of the cube, it's the surface area of one face of the cube multiplied by 6, because there are 6 faces on the cube. So if the cube has a dimension of 1 unit on edge, then the surface area of one face is 1 times 1, or 1 square unit. And then we multiply that by 6 to give the surface area. That would be 6 square units. The volume, however, is the length by the breadth by the height. And seeing as how each edge is 1 unit in length, that would be 1 times 1 times 1, which is 1 cubic unit. And so the surface area to volume ratio is 6 divided by 1, or 6 over 1 if you want to put it as a ratio, 6 to 1. If we then take a large cube, which would be the equivalent of the eukaryotic cell without any internal compartments, no internal membranes, and we increase the dimension of the edge by a factor of 5, so the edge is now 5 units long rather than 1 unit long, then the surface area is still the surface area of the face of one face of the cube multiplied by 6. So one face has a surface area of 25, 5 times 5, and then there are 6 sides, so 6 times 25 is 150. And you can figure out the math, it's pretty easy. The volume is still length by breadth by height, So that would be 5 times 5 times 5. So 5 5s are 25. 5 times 25 is 125. And when you do the math now, the surface area to volume is 150 divided by 125, and that's 1.2. So the surface area to volume ratio has gone down as the cube gets bigger. And the same is true for any shape, whether it be a sphere, whether it be a cylinder, which would be more representative of a cell. The surface area increases by the square, but the volume increases by the cube. So the volume increases much faster than the surface area increases. And so as a shape, a three-dimensional shape gets bigger, the volume increases faster than the surface area. So as any three-dimensional shape gets bigger, the surface area to volume ratio decreases. And this would mean that as a cell got bigger, the ratio of the surface area of the cell membrane, the outer membrane or the plasma membrane, compared to the volume of cytoplasm on the inside, would decrease. And that's important because the membrane determines what can move into and out of the cell. And the rate at which material can move into and out of the cell and how much material can move into and out of the cell determines how much cellular chemistry can occur in the volume inside the cell. And so by seeing the surface area decrease with respect to the volume of the cell as the cell gets bigger, you are putting limits on the cellular chemistry. So how can you allow a cell to get bigger and still maintain a constant or a near constant surface area to volume ratio? And as you might guess, it's the solution that eukaryotic cells have. Divide the inside of the cell into separate compartments surrounded by membranes. And then sequestering or isolating certain parts of the cellular chemistry into those compartments, the organelles, so that they can specialize in particular types of cellular chemistry and therefore become far more efficient and effective at doing that chemistry. And again, we can see this reflected in the surface area to volume ratio. So now if we take our 5 cubic unit cube and subdivide it into smaller cubes, each of 1 cubic unit, or to think in terms of the size of one side of the cube, 1 unit, we see that we've restored the surface area to volume ratio, because now instead of having 1 cube with a surface area of 150, we've actually got how many cubes in one cubic unit cubes in there? We've got 125 of them in there. And so it's now 125 times 6 is the surface area, and 125 times 6 is 750. That's the total surface area of all 125 1 cubic unit cubes inside this larger cubic mass. The surface, the volume is still 125 cubic units because we haven't changed the effective size of this cube. We've just subdivided into smaller cubes. So the volume is still 125. So now the surface area to volume is 750 over 125, and that's 6. We've restored the surface area to volume ratio. So increasing the compartmentalization, subdividing a large cell into smaller spaces, is the key to maintaining a good surface area to volume ratio. We don't have the perfect subdivision in a eukaryotic cell. As a eukaryotic cell gets bigger, we don't retain the same surface area to volume ratio through subdivision, but we do improve the surface area to volume ratio by subdivision, which allows eukaryotic cells on average to get much bigger than a prokaryotic cell and still be chemically as flexible, as effective, and as efficient as the smaller prokaryotic cell. So there are very real benefits for a cell to have subdivisions, and it's the reason why eukaryotic cells have them. So when we start looking at the two major types of eukaryotic cells that we will be considering this semester, we need to take into account the similarities and the differences between plant cells and animal cells. We could go on, we could also talk about fungal cells, and we could talk about protist cells, but there we're talking about cells that are somewhat different, particularly in the case of protist cells. There is such a wide variety of protists that it's very difficult to come up with a standard protist cell. And the same is somewhat true for fungal cells as well. However, if your intention is to go on and take the General Biology 2 class in another semester, then you will delve into those organisms and the structure of those cells as well, because we cover everything in the General Biology 2 class. So we do look at fungi, we do look at all the wide variety of protists, but we also focus on plant and animal cells as well. But in this class, we're just going to focus on plant and animal cells. So the first thing that we recognize when we look at an animal cell is that the only thing that surrounds the outside of the cell and demarcates the cell is the plasma membrane. There is a loose arrangement of material that lies outside of the plasma membrane called the extracellular matrix, but it is not a cell wall, nor should it be considered or construed as a cell wall. It is not rigid, it is quite flexible. It does not prevent the cell from swelling up and bursting, which it could very well do. And when you do the 2 diffusion labs, in upcoming weeks, you'll see examples of what can happen to an animal cell when it's confronted with an environmental situation where water can move into it. It's not pretty. Let me assure you of that. It's not pretty. So when we start looking at the animal cell and we move inside, of course, we're going to find the cytoplasm. We're going to find ribosomes, because we know that those are universal features. We're going to find DNA. It's going to be inside the nucleus, because that's the defining structural feature of all eukaryotic cells, the presence of the nucleus. But then we also expect to find other features. So we find endoplasmic reticulum, otherwise known as ER, and that is subdivided into 2 types of endoplasmic reticulum, rough and smooth. Rough is folded layers of membrane. It's described as rough because attached to the surface of the membrane are ribosomes, giving it a very rough appearance, because you see these blobs of ribosomes on there. And what is also important to recognize, and this picture shows it, but you'll see a more detailed picture, the endoplasmic reticulum and the double membrane that surrounds the nucleus, the plasma membrane or nuclear membrane or nuclear envelope, is continuous or contiguous with the endoplasmic reticulum, specifically the rough endoplasmic reticulum. Then you have the smooth endoplasmic reticulum, which is more tubular-like in appearance, rather than folded membrane. And each part of the endoplasmic reticulum serves a specialized purpose, which we'll talk about when we get there. Also, in terms of folded membranes that look very similar to rough endoplasmic reticulum are the Golgi apparatus, which function as a cellular warehouse and storage location. You will also find transport vesicles, membrane-bound spherical arrangements that carry material from one location to the next. You'll find some specialized versions of the transport vesicles known as peroxisomes and lysosomes. We'll talk about them more when we talk about transport vesicles. Then there are the powerhouses of the cells that produce the cellular energy, the mitochondria. We have a framework, an internal framework that holds everything in place and allows things to move around, called the cytoskeleton, made up of 3 units called microtubules, which are the specific parts of the cytoskeleton that act as a guideway for things to move around. Microfilaments, which are in part responsible for cells changing shape and also maintain some of the rigidity of cells, as rigid as an animal cell can ever be. And then intermediate filaments, which are considered to be somewhat unique to animal cells, although there is a suggestion that they are found in plant cells as well. But they were first identified in animal cells just recently, within the last 30 years or so. And it's not entirely clear whether they are a major constituent of plant cells, but they are certainly found in animal cells. Then you are also going to find flagella. And remember, these are very different from the flagella that we see in bacteria. There is a smaller feature that is associated with the flagella. They are called cilia. These are extensions of the cytoskeleton. They really are extensions of the cytoskeleton. And you're going to see those as extensions of the cytoskeletons. Also associated with the cytoskeleton and to some degree with the flagella is a structural feature called a centrosome, which contains a structure called centriole, and you can see one here. These become particularly important in cell division, and very specifically in animal cell division. And when we talk about cell division in chapters 12 and 13, you're going to see the role of the centrosome and the centrioles in more detail there. So these are the basic features that you expect to find in animal cells. When we take a look at plant cells, the first thing that becomes really obvious about a plant cell is that it's surrounded by this rigid cell wall made out of primarily cellulose. And it limits how far a plant cell can swell. And it serves the same purpose as the cell wall on a bacterial cell, stopping the cell from swelling up and bursting, because the plant cells are in an environment which is more or less water, not much of anything else. You're also going to find some features that are fairly, or that are reminiscent of the animal cell, because they are in common. So you are going to find a plasma membrane, you're going to find cytoplasm, you're going to find a nucleus, you're going to find rough and smooth endoplasmic reticulum, you're going to find ribosomes, you're going to find a cytoskeleton with microfilaments and microtubules. Notice no mention of intermediate filaments, because we're not entirely sure that they are uniformly part of plant cells. You do find peroxisomes, there are transport vesicles, smaller in number. There are lysosomes, again, smaller in number than found in plants, in animal cells, but they are present. We do know that they exist. There's a Golgi apparatus. You find mitochondria, because plant cells need to make energy just like animal cells. But then there are some features that are uniquely plant cells and not found in animal cells. So notice no flagella, no cilia, because they're not found in plant cells. And therefore, no centrioles either, because centrioles seem to go along, excuse me, with the presence of cilia and flagella. And given that plant cells don't have cilia and flagella, no centrioles. There is a story there. We might get into that a little bit later if we have time. Maybe not. We'll see. There is a central vacuole. Now, animal cells do have vacuoles, but they're tiny little vacuoles compared to this very large central vacuole in a plant cell. It actually dominates the interior space of the plant cell, very much so. And it has several purposes that we'll talk about when we talk about the vacuoles a little bit later. Then, of course, perhaps the most unique organelle that's found in plant cells is the chloroplast, the site of photosynthesis. And these are found in any cell that carries out photosynthesis. So those would be plant cells and algae cells. And their origin is kind of interesting. And we'll talk about that when we talk about mitochondria and chloroplasts, because the story behind their origins is pretty much the same. And it is considered to be a theory these days. So it is a pretty convincing story, because there's a lot of evidence to back it up. So, number of things that are uniform in both plant and animal cells: the nucleus, the endoplasmic reticulum, both rough and smooth, cell membrane, ribosomes, Cytoskeleton, slightly different composition, but cytoskeleton nonetheless. Mitochondria, specialized vesicles in the form of peroxisomes and lysosomes and transport vesicles. All of Golgi apparatus, all of those are found across the board in both plant and animal cells. What are unique features of animal cells? Well, cilia and flagella, the intermediate filaments, currently as part of the cytoskeleton. Centrioles in the centrosomes in animal cells, and perhaps lysosomes to some degree. I mean, people go backwards and forwards about the nature of lysosomes, whether they are strictly animal cells or whether they're found animal cell components, or whether they're found in animals, in plant cells and other cells as well. I ascribe to the latter idea that lysosomes are actually found in plant cells because I've seen them in electron microscopes. I'm a plant biologist by training, a plant physiologist specifically, and I used to do a lot of work with electron microscopes looking at plant cells as part of my research. And I can tell you that there are lysosomes in plant cells. I've seen them. I know that they exist. So I find it very hard to argue that they don't exist. What are unique to plant cells? Well, obviously the chloroplast, the cell wall, the little connections between plant cells called plasmodesmata that penetrate through the cell wall so plant cells can talk to each other. Those are all features that are uniquely plant cell, and of course the central vacuole. And it's not a bad idea, in fact, to make up a table. I often have students do this as part of an assignment, which I'm not asking you to do this semester. I thought about it. I've decided not to include it, and I'll announce that because I think in the interest of time and just the workload, there's a lot that it's just a lot more work that perhaps you don't need to do. But I'll put the worksheet up so that you can play with it, because it might help you make sense of the features of a cell if you work through it. But it helps to make up a table where one column is the features of a plant cell that are unique. On the other side is a column that has the features that are unique to animal cells, and then a middle column, so 3 columns in your table, and in the middle column, all the features that are common to both plant and animal cells. And if you draw up that table from memory, it helps you solidify your thinking about the 2 different types of cells, and it helps you put into place those features that are unique versus those features that are common. That's one of the things that students struggle with as they start to come to terms with these 2 different types of eukaryotic cells I've discovered. So these give you some examples of different types of eukaryotic cells, obviously plant and animal cells, fungal cells, and the protists. So this is a member of the old protist group. It's actually a single-celled algae called Chlamydomonas. So it's a photosynthetic algae, but it's a member of that big group of confusing organisms that we refer to generally as protists. So quick concept break, because we've been going for a little while now. So briefly describe the structure and function of the nucleus, mitochondrion, chloroplast, and endoplasmic reticulum. Give you about 30 seconds to think this one through before we address this. If you're taking a break, of course, put us on pause, come back again when you're ready. Okay, so let's talk about these organelles very quickly. The nucleus is the site of the genetic material, the DNA. So that's where the DNA is stored. That's where the messenger RNA is going to be produced. It's also where some of the constituents of the ribosomes are produced before they're exported out of the nucleus to the cytoplasm. Mitochondria and the chloroplast are both involved in energy processing. Mitochondrion is where the major energy producing reactions that produce ATP in cell respiration occur. In chloroplasts, it's the reactions associated with photosynthesis, where light energy is harvested to provide the energy to convert water and carbon dioxide into organic molecules. The endoplasmic reticulum, that is the location of much of the biochemical synthesis that occurs in cells. So sugars are produced here, many proteins are produced here, lipids are produced here. It is a major manufacturing site where many of the macromolecules are produced. It's also where a lot of large toxic molecules are broken down in the endoplasmic reticulum. So you're going to find a lot of endoplasmic reticulum in cells that specialize in detoxing organisms. So you find the breakdown of alcohol and drugs occurring in the liver within the endoplasmic reticulum of liver cells, and to some degree in kidney cells as well. So that's what these organelles do. So let's pick up the story again and start looking at some of the organelle systems that we can identify. The good news is that as soon as we start looking at the organelles in a typical eukaryotic cell, we find that they break down into 4 really convenient groups. So the first group is the manufacturing group, and that includes the nucleus and the endoplasmic reticulum, transport vesicles, and the Golgi apparatus. These are sometimes combined together to form a system called the endomembrane system. Sometimes the breakdown organelles are also included in that, so I'll put a dotted line on that one rather than putting a firm line in there. But the breakdown organelles include specialized forms of the transport vesicles, which is why they sometimes get incorporated into the endomembrane system as well. So you have organelles like the peroxisomes and the lysosomes that are included in the breakdown organelles. And we'll see those as we make our way through the endomembrane system. Then you've got 2 organelles that are very specifically associated with energy processing. Obviously, the mitochondria and the chloroplast are the organelles here, but there's a third one that can be incorporated in here too, the peroxisome. So while we can talk about the peroxisome as a breakdown organelle, it also can be incorporated into the energy processing group as well, and many times is, as you'll see. And then there's a catch-all group, basically all the organelles that don't belong in the other 3 groups, which we typically look at as support, movement, and communication. So this is the cytoskeleton, the cilia and the flagella, and the connections between cells, both animal cells and plant cells, that allow the cells to talk to each other and to attach to each other. So we're going to work our way through each of these groups of organelles in sequence, because it's a convenient way of looking at it. And we'll stop after each one for a quick concept check before we move on again. So we're going to address the manufacture and the protein group, the manufacture and the breakdown group together, because they are often linked together, as I said, either completely within the endomembrane system or partially within the endomembrane system. So the manufacturing group most strictly consists of the nucleus, the ribosomes, the endoplasmic reticulum, both rough and smooth, and the Golgi apparatus. So let's look at them. So the nucleus, of course, is where the DNA is located. Typically, it's invisible unless you use very high magnification in an electron microscope, and then you can see the thin threads of DNA inside the nucleus. This form of DNA is called chromatin. And we'll talk a little bit more about the packaging of DNA when we talk about cell division later in the semester. But the chromatin is a very thin thread-like form of the DNA complexed with proteins called, oh, now I've forgotten the name of the proteins. histones. If I have a little bit of time, I can usually think of it. They're called histones. So these are the proteins that are uniquely found in the nucleus that are used to partially fold the chromatin up so that you reduce the length of the DNA molecule so they can be packaged more conveniently into the nucleus. During cell division, we want to hyperpackage the DNA so that it is more convenient to move the DNA around during the early stages of cell division. And so the DNA coils up into a very tightly packaged material that becomes visible in the cell at the time that the cell begins cell division. And at this point, we refer to that as the chromosomes. So while the DNA is invisible, even though they are strictly chromosomes, we don't use the term chromosome, we refer to the DNA as being in the chromatin form. But once it coils up into a form that is visible in the light microscope, because this is all relating to light microscope observations, once it coils up so that it is visible in the light microscope in the nucleus, then we change the designation from chromatin to chromosome. Also within the nucleus, you're going to see a structural feature called the nucleolus. There could be one or there could be multiple nucleoli within the nucleus. This is where the ribosomes are produced, or where the constituents of the ribosomes are produced. They are assembled into their particles. There are two particles, a small particle and a large particle, in the cytoplasm, but the constituents of those particles are created within the nucleolus inside the nucleus. So here you can see an electron microscope image of a nucleus, and you can see the nuclear envelope. It's made up of two membranes, an inner and outer membrane separated by a gap. And that gap is spanned in locations by a complex of proteins that forms a nuclear pore. These are the gatekeepers to the nucleus. These allow some things into the nucleus, they allow some things out of the nucleus, but they specifically prohibit random crossings into the nucleus. They are the gatekeepers, they are filters. So they do allow some things in, they allow some things out, but they do regulate what can move into and out of the nucleus. So you can see the nuclear pore complexes here embedded in the nuclear envelope. So here you can see the chromatin form of the DNA, and these are the histones. And you can see that the DNA wraps around the histone complexes. This reduces its packaging, and then when it is folded up, it starts to coil up into a more complex form, which in turn coils even more. We refer to this as supercoiling, and it's a way of hyperpackaging the DNA to form the chromosomes. The other thing that you should also notice here is that the rough endoplasmic reticulum, you can see, is connected to the double membrane of the nuclear envelope. We're going to see another picture that reinforces this as well. The ribosomes themselves then are the machinery that assemble the proteins. They come in two forms, what is referred to as free ribosomes and what are referred to as bound ribosomes. The bound ribosomes actually start out as free ribosomes. So we'll put in here start as free ribosomes. They will attach to the rough ER, and what determines that they're going to attach to the rough ER is the proteins that they're going to be making, or the polypeptides that they're going to be making. So free ribosomes in the cytoplasm or cytosol are making polypeptides that specifically are not going to enter the endomembrane system that we're starting to talk about as we start to talk about these manufacturing organelles. Any polypeptide that's going to enter the endomembrane system begins that journey by entering the endoplasmic reticulum, specifically the rough endoplasmic reticulum. So if a ribosome is making a polypeptide that is never going to enter the endomembrane system, then the ribosome remains suspended in the cytoplasm. If, however, that polypeptide needs to enter the endomembrane system because of the journey that it needs to go on to get to its final location, then the ribosome complex is carried down to the surface of the rough endoplasmic reticulum, where it attaches to the surface. And it's the presence of these ribosomes on the surface of the rough endoplasmic reticulum that has led to that name, RUF-ER. And it's because these ribosomes are making these polypeptides that need to enter the endomembrane system that results in the ribosomes attaching to the surface of the RUF-ER. So here you can see the ribosomes in the cytoplasm, but then if you look along the surface of the rough endoplasmic reticulum, which is what these membranes are, you can see the ribosomes attached to the surface. So the ones out here are the free ribosomes making polypeptides that are not going to enter the endomembrane system. These ribosomes attached to the surface of the RUF-ER are making polypeptides that need to enter the endomembrane system. And you can see here the two subunits, the large subunit and small subunit. We'll go into more detail about these when we get to chapter 17 and we talk about the actual process of translation, which is the process by which polypeptides are assembled. And so we'll talk more about the role of ribosomes when we get there. So the endomembrane system, as I said, is the site of much biochemical synthesis. So sugars are produced there because one of the most common alterations to polypeptides is the addition of sugars to form what are called glycoproteins. So those sugars have got to be made somewhere and they're made in the endoplasmic reticulum, specifically the smooth endoplasmic reticulum. All of the lipids that are involved in all of the membranes, wherever they are located within cells, but particularly the membranes of the organelles that are involved in the endomembrane system, are made in the smooth endoplasmic reticulum. And there's a reason for that, as you'll see. The smooth endoplasmic reticulum, as I mentioned earlier, detoxifies drugs. It breaks down alcohol. It breaks down other chemicals that are considered to be toxic or poisonous. It helps eliminate the nitrogen or the amine groups attached to amino acids if those amino acids are going to be used as an energy source, because the nitrogen of the amino groups are toxic to the enzymes of cell respiration. So it's within the liver that those toxic amino groups are removed and ultimately secreted through the kidney. It also serves as a store of calcium. It says here in muscle cells, but also in animal egg cells. We don't really talk about that role in this class, but again, if you take the General Biology 2 class, we do a chapter on embryological development, which begins with the fusion of egg and sperm, fertilization, and the role of calcium becomes really important there because it activates the egg cell. And that calcium comes from the smooth endoplasmic reticulum. So the smooth endoplasmic reticulum is a storehouse, but for specialized storage and also for chemical synthesis or biochemical synthesis, and to some degree breakdown as well. The rough endoplasmic reticulum does one thing and one thing particularly well. It makes polypeptides and proteins through the ribosomes that are attached to the surface. Those are then going to be packaged into vesicles that bud off the rough endoplasmic reticulum called transport vesicles. Those proteins that need to be in the membranes are going to go into the membranes of the rough endoplasmic reticulum and be incorporated into the membranes of the transport vesicles. Those proteins that need to be in the cytoplasm outside of the membranes are going to be in the internal space of the endoplasmic reticulum, what is often referred to as the lumen. Lumen's just a fancy name for a hollow space. inside something, typically surrounded by a membrane. So your stomach has a lumen, that's the hollow space inside the stomach. So these proteins are going to enter the lumen of the rough endoplasmic reticulum and be packaged into the hollow space inside the transport vesicle. So here you can see the rough endoplasmic reticulum and the smooth endoplasmic reticulum. I gave you a description of those earlier, but the rough endoplasmic reticulum is folded and contiguous with the nuclear envelope. It's got the ribosomes attached to the surface. The smooth endoplasmic reticulum is more tubular. It is an extension of the rough endoplasmic reticulum, so the two protein systems are continuous. And then you can see the transport vesicles budding off the external surface of the rough endoplasmic reticulum. The term transitional ER is a relatively new term. Again, I'm not sure how consistently this is used, but the textbook has it here. I've typically learned, again, I've been doing this for a long time, but I learned that there was no such thing as transitional ER. We didn't have that term when I learned this. So the transport vesicles just budded off the rough endoplasmic reticulum. Again, we have the name transitional ER here, so we can use that term, and I'll try to remember to use that term. But if I don't, just remember that transitional ER is, at least as far as the book is concerned, a recognized third form of ER, but it is basically just a specialized part of the rough endoplasmic reticulum. So here you can see electron microscope images of both the rough endoplasmic reticulum with its ribosomes and the smooth endoplasmic reticulum extending off that as a more tubular, irregular structure. Then, like all good manufacturing plants, not only do you have a place where the blueprints are stored, that would be the nucleus or the office. Not only do you have manufacturing machinery, which would be the ribosomes, not only do you have a shop floor where the manufacturing machinery is located, which would be the cytoplasm and the rough endoplasmic reticulum, and to some degree the smooth endoplasmic reticulum as well, you also need a warehouse to store things where you can keep them until they're needed, or you can gather them together to make a specialized shipment. You could even modify things if modification is necessary. And the cell is no different. If we're thinking of it as a manufacturing plant, then we need a cellular warehouse, and that would be the Golgi apparatus. The Golgi apparatus looks a little bit like the endoplasmic reticulum in that it is folded sheets of membrane. And so you can see an electron microscope image of a Golgi apparatus, or a Golgi body is another name for it. Why is it called Golgi? Because an Italian anatomist was the first person to describe this, and his name was Golgi. And so these are called Golgi bodies or Golgi apparatus because Golgi was the person who found them. So sometimes we still do carry the names of people who found things. We try and get away from that a little bit, as you'll see in a couple of other chapters a little bit later on in the semester. But here's a situation where we still use the name of the individual who discovered these, and he discovered these several hundred years ago. So this is not a relatively new terminology, but we still use the name. So Golgi discovered these, so they're known as the Golgi apparatus or the Golgi bodies, and they are the cellular warehouse. And like all warehouses, there is a receiving dock and there is a shipping dock. Where the receiving dock is where things come in, the shipping dock is where things go out. So there are two sides to the Golgi apparatus. One side known as the cis, and you may recall, hopefully, from the last presentation where we talked about cis-trans isomers. Cis means same side. So the cis side of or receiving side of the Golgi apparatus is on the same side as the nucleus and the endoplasmic reticulum. So this is the side that faces inwards to the center of the cell. The shipping side is the trans side because it's on the opposite side to the nucleus. It faces towards the plasma membrane, which is by and large the ultimate destination of most of the material that passes through the Golgi apparatus. Not all of it, but most of it is destined to go to the plasma membrane at some point in the life of the cell. So the transport vesicles from the rough endoplasmic reticulum arrive at the Golgi apparatus and merge, because it's a lipid membrane merging with a lipid membrane. So they fuse together to become incorporated into the folded membranes on the receiving side of the Golgi apparatus. This deposits the polypeptides and proteins in the membrane and in the hollow space of the transport vesicle into the membranes and the hollow space of the Golgi apparatus. Things then move through if there are going to be modifications like adding a sugar, or polypeptides are going to be grouped together to form protein complexes. A lot of this is going to take place here in the Golgi apparatus. Eventually, they're going to be repackaged into more transport vesicles that are going to ship them onwards from the shipping face of the Golgi apparatus onto their final destination, which, as I indicated earlier, is most likely to be the plasma membrane, but may not always be the plasma membrane. So that's the Golgi apparatus. So we now need to take a quick diversion to talk about some specialized forms of transport vesicles known as lysosomes and peroxisomes, because peroxisomes can be included in this. So lysosomes are specialized transport vesicles that contain digestive enzymes. So these are enzymes that would break down sugars, would break down proteins, would break down fats. These are for waste disposal. And waste disposal comes in a number of different forms. So waste disposal could be a way of disposing of organelles inside cells like mitochondria and other organelles that have now become corrupted and can't do their job properly. There's too many good molecules in those organelles to throw those away. So the lysosomes encapsulate those and digest the molecules down to their various components to recycle them back into the cell so they can be used to create more useful things for the cell. Rather than throw things away, just recycle them. And so that's one of the useful features of a lysosome. And cells have been recycling materials pretty much as long as they've been cells. Even prokaryotic cells recycle materials, but eukaryotic cells have specialized organelles to do this. There are some specialized types of breakdown, however, that lysosomes can perform. Phagocytosis is a way of getting rid of external waste, I guess, is probably a good way of putting it. So I probably should have shown you this a little bit earlier. So you can see the lysosomes here in an animal cell in this case, and you can see that the lysosome has fused with a mitochondrion. There's a peroxisome in here as well, and these are being digested down to recycle them because they're damaged. They're no longer functional, so they're damaged and they're being disposed of. The peroxisome is helping in the breakdown as well. If you've got phagocytosis going on, then this is the capture of a food particle, or it could be a bacterium in the case of cells from the immune system. They get encapsulated in a specialized vacuole called a food vacuole. A lysosome then comes along and fuses with it. The digestive enzymes go to work and break this down. So there are cells that you will encounter along the way this semester called amoebas. These are single-celled protists, and they feed this way. This is how they feed. And there are actually lots of unicellular protists that feed in this fashion by capturing food particles and then digesting the material inside the food particle using the enzymes of lysosomes. And then autophagy is simply the breakdown of damaged and non-functional organelles using lysosomes again. So those are the lysosomes. Then we've talked about the food vacuoles and specialized vacuoles. We will talk about the central vacuoles in plant cells in a little bit more detail in a moment. But there's another specialized type of vacuole that you're going to discover in protists. We'll encounter these again in chapter 7 when we talk about regulating water flow into and out of cells. And this is called a contractile vacuole, which is actually a water pump. It's one way of controlling the flow of water into cells. So because the vast majority of cells do live in an environment where water is typically going to flow into the cell, you've got to be able to regulate that, because again, if you don't, the cell will swell up and burst. Not a good thing. So we've seen the role of a cell wall in doing this, but another way of dealing with this is to let the water come in, but to capture all of that excess water and simply pump it back out again. And that's what many protists do. And you'll see an example of it in chapter 7. And there is even a posted video showing a contractile vacuole in process of collecting and pumping water out. And so these contractile vacuoles swell up as they capture water, and then they collapse down very suddenly to pump the water out, and then they start swelling back up again, and then they collapse to pump the water out. And they collect water from throughout the cytoplasm to pump it back out of the cell again. So it's a way of regulating water flow. And then plant cells have these large central vacuoles, and you can see here in this electron microscope image what I was referring to when I said this thing dominates the internal space of a plant cell. Here's the nucleus, here are the chloroplasts, here's the central vacuole. And typically, the central vacuole is the single largest organelle inside a plant cell. And it can do a number of different things. So it can accumulate water. So, writing on the wrong screen. So water can move in and out. And that has an important role because the water that moves in and out causes the central vacuole to swell and collapse depending on how much water is in there. When it swells, obviously it's going to put pressure on the inside of the cell, which is going to push the inside of the plant cell hard up against the cell wall, a condition called turgor. And we say that the plant cell is turgid. And this is what holds your houseplants in a rigid condition. And you know what happens when you fail to water your houseplants frequently enough. They start to wilt. And what's happening is that the water content of the central vacuole is decreasing because there's not enough water to fill the vacuole. And that's what causes the leaves to wilt. And you know how to fix that. Just rewater it. That refills the central vacuole. It swells up. And the cell wall simply limits how far the central vacuole can swell, and therefore how far the plant cell can swell. But the typical condition of a plant cell is to be rigid or turgid because of this pressure of water. We'll talk more about that in chapter 7. What else can the central vacuole do? Well, it can store waste. Because a plant cell has no way of releasing its waste like an animal does, nor can it get up and walk away from it if it could. It stores a lot of the more toxic waste inside the central vacuole where it can do no damage to the cellular chemistry. It can also be a storage place for all sorts of good things. So when you drink a glass of orange juice, you're actually drinking vacuole juice from an orange. And that vacuole juice contains sugars, flavorings, colorants. These are all naturally occurring chemicals that are produced by the orange, in part to give the orange juice a nice flavor so that the orange itself has a nice flavor, so it's a little bit more edible to animals. Why? Because an orange is a fruit, and even though the oranges that we typically eat are navel oranges without seeds, and that's because of a mutation, the classical orange would have seeds in it, the classical citrus would have seeds in it, and the fruit, whether it be a lemon or a lime or an orange, would be used for seed distribution. And you might find that rather puzzling given how bitter some of those citrus fruits can be, but animals that would specialize in eating citrus fruits can tolerate those bitter chemicals. And basically that's a way for the citrus to limit who does and who doesn't eat their fruits. So the specialists who can most easily distribute the citrus seeds to the benefit of the citrus can eat the fruits, and those that are not going to do it properly or do it efficiently won't eat the citrus because it's just not palatable. We've just figured out that we can strip the unpalatable part, the skin, off the outside, and as long as we wait for the citrus to sweeten up, which it does as it changes color from green to orange, and it changes its smell so that it smells attractive, we can strip the skin off and we can enjoy the inner part of it where all the goodness is, the sugars and the other things. But again, the orange juice is coming out of the central vacuole. So a cup of or a glass of orange juice in the morning, you're drinking vacuole juice from the orange. So when we start thinking in terms of the endomembrane system as a series of organelles linked together by transport vesicles, we start to understand the reason why certain things are associated with the endomembrane system. So number one, we know that the endomembrane system is involved in protein synthesis. That's the role of the ribosomes attached to the rough ER, which is a central part of the endomembrane system. Also, the nuclear envelope has ribosomes attached to it. And remember, the nuclear envelope and the rough endoplasmic reticulum are a continuous membrane system. So no problem there with the proteins getting transported. But we also need to recognize that the lipids that make up the transport vesicles are being extracted out of the rough endoplasmic reticulum. So if they weren't replaced, forming these transport vesicles, and that we would have to include there the peroxisomes and the lysosomes and other specialized vesicles that are formed from the endoplasmic reticulum. If we didn't replace all the lipids that were involved in forming those, we'd very quickly run out of lipids. So the smooth endoplasmic reticulum makes the lipids that are used to replace the lost lipids when the transport vesicles are formed. So given that the smooth endoplasmic reticulum, the rough endoplasmic reticulum, and the nuclear envelope form one large membrane system, it stands to reason that the smooth endoplasmic reticulum is making the lipids for all of those. So that also explains why the smooth endoplasmic reticulum is involved in lipid synthesis. But wait, there's more. Because those transport vesicles carry their lipids to the Golgi apparatus. So the lipids that are being extracted out of the endoplasmic reticulum now form part of the Golgi apparatus. But wait, there's more. Because eventually, remember, the proteins are going to be repackaged into transport vesicles. So the lipids that came in as transport vesicles are now going to be extracted as transport vesicles. Where do those transport vesicles go? Well, they become the lysosomes and peroxisomes, or in the case of many of them, or in fact, most of them, they come on out and fuse with the plasma membrane. So the lipids of the transport vesicles become incorporated as lipids in the plasma membrane. And any proteins in the membrane of the transport vesicle become proteins in the plasma membrane. And any proteins in the hollow space of the transport vesicle get released out of the cell. So the lipids of the plasma membrane trace their origin through the transport vesicles, through the Golgi apparatus, through the transport vesicle, through the rough endoplasmic reticulum, all the way back to the smooth endoplasmic reticulum. So now we start to get a feel for why the endoplasmic reticulum, both rough and smooth, must be considered as a major biochemical manufacturing site within the cell. Smooth endoplasmic reticulum is making lipids for all of these membranes and elsewhere as well. Mitochondria and chloroplasts have membranes. And those have the lipids have to come from somewhere as well. And the mitochondria and chloroplasts don't make their own lipids. So it falls back on the smooth endoplasmic reticulum again. Of course, the rough endoplasmic reticulum is making proteins. Remember that in the Golgi apparatus and also to some degree in the rough endoplasmic reticulum, many proteins are modified by the addition of sugars to create glycoproteins. Those sugars have got to come from somewhere, so they are made by the smooth endoplasmic reticulum. So again, more biochemical manufacturing. The digestive enzymes of the lysosomes, well, they're proteins. They are made by the ribosomes of the rough endoplasmic reticulum. So if you're getting this idea that the endoplasmic reticulum and the endomembrane system in general is a major biochemical manufacturing plant as far as the cell is concerned, that's really good, because that's exactly what it is, and that's exactly how you need to think about it. And the various components are tied together by these mobile units, the transport vesicles, that link the nuclear envelope endoplasmic reticulum manufacturing plant with the warehouse, the Golgi apparatus, which in turn is linked to the final destination, the customer, if you wish, the plasma membrane. And the transport vesicles should be considered as the trucks that transport materials from one location to the next, linking all of these features together and integrating this into a single manufacturing system inside the cell. Very, very important part of the cell. So, time for a quick break again. And again, the concept checks relate back to what we've been talking about. So if you want to take a quick break, bathroom break, get a drink, get a snack, by all means, put me on pause. I'll still be here when you come back. Otherwise, let's read through the questions. I'll give you about 30 seconds or so to think about it, and then we'll look at the answers. So first one, differentiate between the structure and function of the rough and smooth endoplasmic reticulum. And number two, how do transport vesicles integrate the components of the endomembrane system? Imagine the Jeopardy music. Okay, welcome back again. Whether you've been on a break, whether you've just restarted the presentation, or whether you stayed with me, let's take a look at the questions. Differentiate between the structure and function of the rough and smooth endoplasmic reticulum. Well, the smooth endoplasmic reticulum, remember, is the tubular form. It's a site of manufacturing for lipids and carbohydrates. It's also for storage of calcium and a number of other things. It's also a site of breakdown of alcohol and drugs and other toxins that need to be broken down in the liver and to some degree in the kidney as well. The rough endoplasmic reticulum is folded sheets of membrane with a hollow space in between the sheets. You have ribosomes attached to the surface that are making polypeptides and proteins that are going to be transported through the endomembrane system. It is the site of the packaging of the polypeptides and proteins into transport vesicles for transport to other parts of the endomembrane system, most notably the Golgi apparatus as an intermediate location, or maybe in some cases directly to the plasma membrane. So how do the transport vesicles integrate the components of the endomembrane system? By moving things from the rough ER to the Golgi, and then moving things from the Golgi to the plasma membrane, and also moving things from the Golgi out into the cytoplasm in the case of the specialized transport vesicles, the lysosomes and the peroxisomes. So if that's how you were thinking, good, you're starting to master the endomembrane system. So now let's address the energy processing organelles. And you can see here that the peroxisomes feature, although we can think of them also as specialized versions of transport vesicles, they are involved in energy processing as well, as you will see. So mitochondria and chloroplasts are usually the ones that we consider when we think of energy processing. Mitochondria associated with cell respiration. Put that in here. Chloroplasts, of course, photosynthesis. And then peroxisomes, well, they do a grab bag of things, but mostly they're involved in fat breakdown. And remember, the fat is a really good source of energy. And we'll see more of that when we do cell respiration. And then a series of reactions that are referred to as oxidation and energy recovery. And that's particularly in plant cells. So let's take a look at the organelles in a little bit more detail. So when we look at mitochondria and chloroplasts, one of the things that struck a biologist named Lynn Margulies back in the early 1970s was just how similar mitochondria and chloroplasts are to bacteria. The double membrane feature of mitochondria and chloroplasts is somewhat unique as far as organelles are concerned in cells. Most of them have a single membrane. And in fact, the double membrane feature of the nucleus is sort of an outlier in terms of what a double membrane is like, because that double membrane is an envelope rather than a true double membrane system that you find in the mitochondria and chloroplast. And so having this double membrane around the outside of them, while it doesn't relate specifically to a bacterial feature, it started to give us cause to think, why do they have this double membrane? And then when you look at the inner membrane of both mitochondria and chloroplasts, it's actually connected to a highly folded membrane system. In the mitochondria, the inner membrane is directly folded into fingers of membrane called the cristae, and sometimes it's referred to as the cristae membrane. And then the inner membrane system of the chloroplast, which is referred to as the thylakoid membrane system, at first glance appears to be separate from the inner membrane of the chloroplast, but in fact, very careful study with electron microscopes indicates that it is in fact derived from that inner envelope. So both of the inner membrane, folded inner membrane material or structures in mitochondria and chloroplasts derive from the inner membrane of the organelles. And when you look at bacteria that carry out cell respiration, aerobic bacteria, and chloroplasts that carry out photosynthesis, known as cyanobacteria, they also have folded extensions of their plasma membrane, their cell membrane, that give rise to structures, membranes, that are attached to their plasma membranes that are involved in cell respiration and photosynthesis respectively, just as we see in mitochondria and chloroplasts. What is more amazing and more supportive of this idea of their similarity to bacteria is that both mitochondria and chloroplasts have ribosomes for producing protein. And they have their own DNA molecules in the form of circular DNA molecules, just like bacteria, that contain genes that are transcribed and translated, and the ribosomes synthesize proteins. And these proteins are involved in cell respiration and in photosynthesis in the mitochondria and chloroplasts, respectively. Those ribosomes are more like bacterial ribosomes. In fact, they're essentially identical to bacterial ribosomes. And there are a couple of antibiotics that will shut down protein synthesis in bacteria, and those same antibiotics will shut down protein synthesis in mitochondria and chloroplasts, reinforcing the fact that these ribosomes are truly bacterial ribosomes. When a plant cell divides, when an animal cell divides, when any cell divides, the mitochondria, which are found in all eukaryotic cells, and the chloroplasts, which are uniquely found in plant cells, don't divide. They divide at a later stage in the cell life cycle, independently of their host cell. So they have a semi-independent existence. We actually refer to them as semi-autonomous. And Margulis suggested that what this indicates is that at some point in the past, both of these organelles were actually free-swimming bacteria in the case of mitochondria, and aerobic bacterium in the case of the chloroplast, a cyanobacterium. And that at some point, a very primitive eukaryotic cell captured an aerobic bacterium, but instead of eating it, preserved it. And that relationship became solidified over time, whereby rather than seeing the aerobic bacterium as a source of food, it was viewed as a source of ATP, to the benefit of the eukaryotic cell that had captured it. Because now it didn't have to go and try and find a source of ATP. It was capable of making its own by hosting this bacterium. And as this relationship became more and more solidified, some of the genes that were associated with the bacterial function were transferred to the nucleus. So there are genes in the nucleus now that are related to mitochondrial function. But the mitochondria still retains some independence because it has its own DNA and the ability to make its own some of its own proteins. At a later point in time, a eukaryotic cell that had a mitochondrion or had mitochondria already captured a cyanobacterium, and again, rather than eating it, created a relationship with this cyanobacterium that became solidified over time in much the same way as the relationship to the aerobic bacterium became solidified. And the cyanobacterium eventually evolved into what we now refer to as a chloroplast. And again, some of the DNA sequences of the chloroplast were transferred to the nucleus of the host cell, so that neither are independent of the other. They're semi-independent of each other, but they rely on each other. And this gave rise to the precursor to algal cells and ultimately plant cells and plants in general. This is referred to as the endosymbiotic theory of the origin of chloroplasts and mitochondria, and it has a substantial body of support. It took, however, over 20 years for the biological world to accept Margulis' idea as having validity and eventually accumulating enough information, enough data that was supportive of Margulis' hypothesis that it would eventually rise to the level of a theory as it is today. And so the way we view the development of eukaryotic cells follows much along this pathway. We can account for the origin of the endomembrane system as infoldings of the original plasma membrane. Because remember, the plasma membrane, the Golgi apparatus, the endoplasmic reticulum, the nuclear envelope, these all form part of that integrated system, the endomembrane system. So it's not hard to imagine how the endomembrane system may have evolved from invaginations of the plasma membrane, because they're all still linked together, and the plasma membrane is still solidly tied to all of the other membranes of the system via the transport vesicles and the proteins being produced on the rough endoplasmic reticulum. But then to account for the origin of the mitochondrion, we have the capture of an aerobic bacterium that's carrying out cell respiration in a similar fashion to the way we see it in mitochondria today, because in terms of the biochemistry of cell respiration, the genetics of cell respiration, it's identical in the aerobic bacteria and in the mitochondria, which is another piece of information that ties the two together. Then at a later point in time, a cyanobacterium gets captured by a eukaryotic cell that already has mitochondria, and that evolves into the chloroplast. And again, the big piece is not just the similarity in terms of looking and behaving like bacteria, but the genetics and the biochemistry of photosynthesis in cyanobacteria and in chloroplasts, wherever you find them, is essentially identical. To all intents and purposes, it is identical. Minor tweaks, but genetically, biochemically, chemically, however you want to look at it, it's identical. And so all of this evidence adds up to supporting Margulies' original contention that chloroplasts and mitochondria were once free-swimming bacteria back in deep, deep time. I mean, this goes way back, at least 3 billion years, maybe even further back into the history of life, where we start to see the appearance, well, yeah, probably 3 billion years, we start to see the precursors of eukaryotic cells in a formal sense around 2 to 2.5 billion years ago, somewhere in that framework. And so the body of information, the body of data that now supports this theory is quite extensive. But as I said, it took a while for people to be convinced that Margulies was on the right track in terms of her thinking about the origin of mitochondria and chloroplasts. But she stuck with it because she was certain that she was correct, even though there wasn't a lot of information when she first proposed this hypothesis. But over time, as more and more people started to think as Margulies was thinking, and data started to be collected on the basis of Margulies being correct, it became really obvious that the data was totally supportive of what Margulies was suggesting, the point now where we refer to this as a theory. So mitochondria, found in all eukaryotic cells, with the exception of a couple of protists. But even those protists have remnants of mitochondria, and there are genetic sequences related to mitochondria in the nuclear DNA, indicating that while these protists no longer have functional mitochondria, and the reason why they don't is because they're parasites, so they don't need them. While they don't have mitochondria, at one point in the past, the ancestral cells to these organisms did have mitochondria, because the evidence is there in the form of the genetic sequences. Those genetic sequences wouldn't be there if they didn't have functional mitochondria. They just discarded them because they don't need them anymore. As I pointed out, it's surrounded by a double membrane. If we look at a picture, the outer membrane is a smooth membrane. You could think of this as being derived from the original food vacuole. It's not a bad that was involved in capturing the bacteria. That's kind of where it may have been derived from. The inner folded membrane is derived from the plasma membrane, and it is this membrane and the two compartments that are defined by it, the inner compartment called the stroma and the space between the inner and outer membrane, that are involved in the reactions of cell respiration that are associated with the mitochondria. So two compartments separated by a membrane is key to understanding how ATP is generated. And you see that structural relationship in the mitochondria. Why the folding of the inner membrane? To create more membrane surface area to carry out the processes, which is the clue that this membrane must be involved in ATP generation. The more membrane, the more ATP you can generate. In the chloroplast, is involved in capturing light energy. Again, it's got the double membrane system. The inner membrane system, as I mentioned, appears to be separate from the actual inner membrane of the chloroplast, but careful study with electron microscopes indicate that the two are connected together. This is the inner membrane of the chloroplast, or the folded membranes of the chloroplast are much more complicated than those of the mitochondria. And this is because the capturing of light occurs on the surface of these membranes. So they are a series of discs called thylakoids. Each disc is connected to all of the other discs in these stacks. A stack is called a granum, the plural is grana. Each grana stack is connected to adjacent grana stacks by connecting membranes, often referred to as lamellae membranes. And so what that means is that the space of each thylakoid is connected to the space of each of all the other thylakoids in the stack, and then the spaces in the stack are connected to the spaces in all the other stacks by these connecting membranes. So you actually have two spaces separated by a membrane inside the chloroplast. The two spaces are the stroma, the equivalent of the matrix or the cytoplasm of the cell, or the matrix of the stroma, the matrix of the mitochondria. Then the second compartment is the space enclosed in all the thylakoids, and the membrane that separates them is the thylakoid membrane. And if that suggests that maybe you could make ATP here, good, because you can. So ATP is one of the products of capturing light energy, and it serves as a source of chemical energy during the conversion of carbon dioxide to organic molecules in the Calvin cycle, which occurs in the stroma. So we start to see, again, a structure-function relationship in both the chloroplast and the mitochondria. And we'll come back and we'll talk about these again in more detail as we talk about cell respiration and photosynthesis. Then we have the peroxisome. It's involved in breaking down fatty acids via a process called beta oxidation, which then disassembles fats into into molecules that contain two carbons, are called acetyl-CoA. And you're going to see acetyl-CoA play a central role in the cellular reactions or the chemical reactions of cell respiration that occur in the mitochondria. So the presence of peroxisomes is an important part of animal cells and also plant cells. Peroxisomes are also involved in breaking down molecules that we've referred to as oxygen-free radicals, an example of which would be peroxide. And the problem for plants is that because they make oxygen as a waste product during photosynthesis, there are always going to be a lot of oxygen-free radicals around. And so typically in a plant cell, it is not unusual to find peroxisomes adjacent to chloroplasts and also adjacent to mitochondria to serve as both a detoxifier of peroxides and other oxygen-free radicals, but also as a way of scavenging back some energy that can be lost through, due to and I'll use the word defect in photosynthesis because of the enzyme that plays a central role in capturing carbon dioxide, a process called carbon dioxide fixation. We'll get into more detail with that when we get into photosynthesis. But this image showing a peroxisome adjacent to chloroplast and to a mitochondrion is not unusual in plant cells because this scavenging process involves transfer of chemicals from the chloroplast to the peroxisome, where a series of chemical reactions occur, which then passes the products on to the mitochondria, where more processing is concerned, and then products from the mitochondria are moved back to the chloroplast again. And this transfer of materials from the chloroplast to the peroxisome to the mitochondria allows the plant to scavenge back some energy that would otherwise be lost because of this central defect in the process of photosynthesis. But more details on that when we do photosynthesis in chapter 10. So let's stop again very quickly for another concept check before we move on to the last group of organelles, the catch-all group: support, movement, and connection or communication. So the concept check question at this break is identify and describe two characteristics, either structural or functional, shared by both chloroplasts and mitochondria. And there are a number of them, but all you need to do is come up with two. Cue the Jeopardy music. Okay, so if you said anything along these lines, double membrane, folded inner membrane, multiple compartments separated by a membrane, an inner membrane, presence of circular DNA, presence of ribosomes, semi-independent behavior or existence, any of those would work. Any combination of two of those would work as an answer to this question. Hopefully you came up with most of those as you were thinking about. So let's get into this last group of organelles so we can finish up for this presentation. So this is our catch-all group. Everything that fits in here doesn't fit into any of the other three groups that we've talked about. So we've got support, we've got movement, we've got communication. And in fact, support and movement are tied together the cytoskeleton, centrioles and centrosomes, the cilia and flagella. So this is support and movement. And of course, the connection between the cells, those are for communication. Because cells in multicellular organisms, at least, do need to be able to communicate with each other. Kind of important. So let's take a look at all of these, starting with the cytoskeleton. So the cytoskeleton, the name is a little bit of a misnomer because if you think in terms of skeleton, you're probably thinking of something that is rigid and inflexible and could be shattered because it's made out of hard material. Nothing could be further from the truth in terms of a cellular cytoskeleton. This is made up of protein. It is flexible, although it does provide some internal rigidity to the cell in terms of helping the cell maintain its shape. But it is also involved in allowing the cell to change its shape. And if you go on YouTube or Google amoeba, or take a look at the video that's posted in the resource section. As I recall, I posted one there. I'll double check to make sure that I did. And if I didn't, I'll put one in there. But if you take a look at any video of amoebas, you'll see that they are constantly changing shape. And that is driven by the cytoskeleton, particularly by the microtubules, which is part of what microtubules do. They not only help the cell maintain its shape, but they are also responsible for allowing cells to change their shape because they are the driving force behind that. So the cytoskeleton is kind of important in that it gives what would otherwise be an irregular shaped cell some regularity to its shape, although in the case of animal cells, they're still pretty irregular in shape, but at least they're not totally irregular and without shape. So here you can see the various components of the cytoskeleton stained with different colored stains. And so you can see microtubules and you can see microfilaments. And I believe, you can also see the intermediate filaments. The different colored stains highlight different components of the cells. So there are three very distinct components to the cytoskeleton, and I've just named them on the previous slide. Microtubules, which are the largest, the microfilaments, which are the smallest, and then the intermediate filaments, which in size lie somewhere in between. And so this table, which is taken directly out of your textbook and is a good one to look at, gives you all of the features of the three components, their size, what they're made up of, what their function is, and then shows you the location of the various components of the microskeleton or the cytoskeleton inside, in this case, animal cells. So you can see the microtubules, the largest component, made up of two polypeptides called alpha and beta tubulin. They form combined units, which are referred to as dimers because they're the two polypeptides together, and it forms a hollow tube that can be assembled and disassembled pretty quickly. And these are responsible for movement. They guide the movement of things, and they are also responsible for maintaining and changing cellular shapes. And we'll see examples of those in upcoming chapters. The actin filaments are contractile. So these are the elastic filaments, and they are contractile elastic filaments that allow muscle cells to contract. So the interaction between actin filaments or microfilaments and another protein complex within muscle cells is what causes muscle cells to contract, which causes muscle fibers to contract, which causes the muscle as a whole to contract. And it's because these fibers, these intermediate or these microfilaments can only contract, that means that you need a pair of muscles around each joint where you can move the appendage in two directions. And so you have a pair of muscles called antagonistic pairs that work against each other. So on your elbow, for instance, you have the bicep, which raises the forearm around your elbow, and you've got the tricep, which lowers the forearm around the elbow, and they work against each other. They form an antagonistic pair. And those of you who are going into our Allied Health Science program or our nursing program and will be taking the ANP classes that we offer, you'll learn more about the role of those muscles and the role of the actin filaments and the other proteins in the muscle cells to allow muscles to contract. You're going to go into deep detail on those because it's really important that you understand how this operates. And then in animal cells, and there is a suspicion that they may be present in plant cells as well, but the presence of keratin suggests maybe not, because keratin is a protein that is pretty much uniquely animal. It's what forms hair, it forms toenails and fingernails and claws and the beaks of birds, the scales on reptiles and that sort of thing. That's all made out of keratin. So the presence of keratin as the protein in intermediate filaments suggests that probably they are going to turn out to be uniquely animal and not present in plant cells at all. But these are the reinforcing fibers. These are the fibers that resist tearing and stretching. So these are the strong fibers of the cytoskeleton. So these are the three components of the cytoskeleton. And so these are talked about in a little bit more detail, and you can read through these slides from the slide set that you can download. I'm not going to talk in detail about these because I've given you information about them as we looked at the table. Here you can see the microtubule and the thing that, and the feature that's moving along it, and it's typically motor proteins that do that. I have some really nice videos somewhere, it's probably in my desk in my office on campus, of things moving around inside a plant cell under the microscope that was provided by a colleague back in Australia that I got when I was still living in Australia and doing research. And you can actually see the organelles zipping along the microtubules. You can't see the microtubules, but you can see the organelles moving along them, and it's quite clear they're following a framework. And then when we start to talk about cell division, particularly when we talk about animal cell division, both in chapters 12 and 13, the two different types of eukaryotic cell division, we're going to see the role of these centrioles. because these form the basis for the development of very important framework structures that form during cell division. Quite remarkably, while these are part of animal cells, the centrioles are part of animal cells, they're not found in plant cells, but the same organizing framework called a spindle that forms in animal cells also forms in plant cells during plant cell division. So it begs the question, why do the centrioles exist? Why are they uniquely found in animal cells? And there seems to be an answer for this, in that when we look at cilia and flagella, and I'm going to jump over a couple of pictures. I think there is a diagram here somewhere. Let me just keep going. Maybe not. No. But in cilia and flagella, so maybe I'll draw it this way. Yeah, so here we've got our diagram of the cilia and flagella. Oh, I lost track of what I was thinking about. It's sitting right here. Look at this cross-sectional structure of a feature called the basal body that sits at the base of every cilia and flagella. Notice it's got what are referred to as triplets of microtubules, and they run lengthwise through the basal body. It's a short little structure. If we go back to the centriole, look at the cross-section here of the centriole. See the triplet of microtubules running the length of this? In fact, one of these structural units within a centriole is a basal body. And these structural features that become the centrioles in your cells, or became the centrioles in your cells, they actually came from the basal body that was contributed by the cilia on the sperm cell that came from your father. It's the only other contribution other than his chromosomes that your father made to you. And those basal bodies become, or this basal body structure becomes the basal bodies of all the cilia and flagella that your cells produce, and then every animal cell produces. But also, by doubling down like this, becomes the basis for the centrioles. And when a cell divides, the spindle that forms has a centriole at each end of the spindle. And when the cell divides, it splits the spindle so that one end of the spindle and its centriole, or the centriole at one end of the spindle, winds up in one cell, and the other centriole at the other end of the spindle winds up in the other cell, to become the basis for centrioles and, I assume, basal bodies in the new cell. It does tie together. I haven't seen our textbook yet discuss all of that, but I've started to see it being mentioned in papers as I do my outside reading. So that seems to be the reason why centrioles exist in animal cells, because they have cilia and flagella that have basal bodies, and they need basal bodies. And the flagellum of a sperm cell in an animal has a basal body, and it is, other than the nucleus of the sperm, the only other part of the sperm that enters into the egg cell during fertilization. And we know it becomes the basis for the centrioles that are involved in cell division. We know that for sure. So there is a connection between basal bodies and centrioles, and I think it goes both ways. So what is the difference between a cilium and a flagellum? Length, basically. The base of both a cilia and flagella wave backwards and forwards, side to side, as a consequence of interaction between the microtubules and some other proteins that make up the flagella and the cilia. In cilia, it means it just waves backwards and forwards. In flagella, the waving backwards and forwards at the base, however, sends this ripple down the flagellum. Some of you may recognize this term, a sinusoidal wave or an S wave that ripples down the flagella, and this is what allows the sperm cell to move in animal cells, or in animals, the sperm cell of animals to move. So when we look at the cross-section of a cilium or a flagellum, they have the same cross-sectional structure. It's called a 9+2 structure. So there are 9 pairs of microtubules. So I'm going to change it to red for the purposes of highlighting the diagram here. So there are 9 pairs of these outer microtubules, and then in the center, there is a single pair. And that's where the 9 plus 2 comes from. 9 pairs plus single. So that's where the 9 plus 2 structure comes from. The inner pair of microtubules are connected to the outer pairs by some spoke proteins, so it looks a little bit like an old spoked wheel. And then the rim of microtubules are connected by motor proteins that are called dynein arms or dynein proteins. And we talked about the structure of the basal body and its connection to centrioles, so we can move on from there. When we look at how the movement takes place, the end of the cilia or flagella is anchored to the basal body. So it's actually the basal body that serves as the anchor. And because these can't move, when the dynein arms move against the microtubules or pull against the microtubules, it causes the whole microtubule array to sway in one direction and then to sway backwards in the other direction, which is why the cilia and flagella at the base wave backwards and forwards. And that's the basis for their movement. And it's ATP energy to transfer phosphate groups to the dynein arms that allow them to pull against the microtubules and relax against the microtubules. It's a similar sort of mechanism that causes the motor proteins in muscle cells, as we'll see in a minute, to pull against the actin filaments, the microfilaments, and it causes the muscle cells to contract. The other thing that I want to point out, and this is not obvious in this picture, but so what you should recognize, you can see the lines pointing to the plasma membrane. So there's the plasma membrane. So you need to bear in mind that the plasma membrane completely surrounds the microtubule array that form the cilia and flagella. So these really should be considered as extensions of the cell proper and extensions of the cytoskeleton, because remember, the microtubules are a central component of the cytoskeleton. So having the microtubule array of the cilia and flagella completely encapsulated inside the cell membrane means that these are cellular extensions. They are enclosed within the cell proper because they're surrounded by the plasma membrane, and they truly are extensions of the cytoskeleton. So then the classic example of the microfilaments is the contraction of muscles. So you can see the protein complex that is the motor proteins. They are the myosin proteins are very similar to the dynein proteins in both appearance and function. So it's the addition and removal of phosphate from ATP to the myosin proteins that allow them to flex against the actin filaments and pull on them. Because the actin filaments are anchored to the inside of the ends of the muscle cells, they pull on the ends of the muscle cells, which causes the cell to contract. And because the muscle cells are anchored to each other in muscle fibers, the whole fiber contracts, which causes the muscle itself to contract. Then, if you're able to see a video of a plant called Elodea, which is something that we use a lot in biology labs. So its name is Elodea. It's spelled E-L-O-D-E-A. But I can see that the transcript service is really struggling with this one. So let me just put this in. So this is actually Elodea here. So Elodea is a little aquatic plant that we use a lot. And when you look at it under the microscope, you can actually see the chloroplast moving around in a racetrack fashion. This is called cytoplasmic streaming, and that is also driven by actin filaments. Likewise, the change in shape of an amoeba, as I said, is mediated by the microtubules, but the microfilaments also play a part because they are the contractile elements. But microtubules have a significant role in helping the cell change shape as well. And we're going to see all of these exemplified in later chapters, all of these functions of the cytoskeleton exemplified in later chapters. Then in terms of the intermediate filaments, we don't talk very much about them because they're a relatively recent discovery. They originally proposed about 20 years ago. They've been formally recognized for about 10 or 15 years. People are still trying to figure out all of their roles, but they are more of an anchoring, resisting stretching, strength component of the cytoskeleton, rather than one that moves things around. And that's because of the nature of keratin and a couple of other proteins that sometimes turn up in them. So those are the movement and support parts of the last group of organelles. So another concept check, just quickly. What are the three components of the cytoskeleton? And describe the 9+2 structure of cilia and flagella. Let's cue the Jeopardy music again. You probably have it as an earworm playing in your head at this point. But let's stop for about 30 seconds. Okay, it might not have been 30 seconds, but let's talk about this one. So what are the three components of the cytoskeleton? The large one is the microtubule, a hollow tube of proteins. The middle one is the intermediate filaments, made up of keratin and a couple of other proteins, but primarily keratin for strength and resisting stretching and tearing. And then the actin filaments, which are made, or microfilaments, which are made up of actin. And those are the contractile components of the cytoskeleton. What's the 9+2 structure? Remember, that is the fact that a cilium or a flagellum has 9 pairs of microtubules in a ring around the outside. So it's 3, 4, 5, 6, 7, 8, 9. So there's the 9 pair, and those are connected by the dynein arms. And then in the center, you have got a pair of microtubules that are connected out to these by the spoke proteins. And that's a consistent cross-section or a common cross-section through both the cilia and the flagella. They have that same shared cross-sectional structure. So let's talk very briefly, very quickly about the final set of cellular structures. what we find outside of the cell membrane, if there is something outside of the cell membrane, and what allows the cells to talk to each other so they can communicate in multicellular organisms. So when we look at plant cells, around the outside of plant cells is a cell wall. The primary component is cellulose, so the primary cell wall is made, ooh, that's not done well. Let me erase that and write it in again. So the primary cell wall, which is the original cell wall that's created when plant cells divide, I don't think that's any better, quite honest, but it'll have to do, is made up of cellulose. Then the secondary cell wall is made up of cellulose, hemicellulose, did that get in? I'm not sure. Yeah, so I can write underneath it. That's interesting. And a third substance called lignin. Now, those are the components of the secondary cell wall. Then the glue that holds them together is the middle lamellae, and that's made up of a substance called pectin. And some of you may recognize the name pectin. This is the material that you use if you want to make homemade jellies and preserves and jams. It's the pectin that interacts or combines with the boatload of sugar that you add to the fruit that you're turning into your jam and causes it to gel and makes the jam or the jelly have that jelly-like consistency. And if the fruit that you're making your jam from won't gel of its own, you can actually buy pectin that's been extracted from plant tissue so that you can make jelly or jam. So the cell wall is a rigid structure around the outside of the plant cell. So plant cells typically have a very regular shape. They look like little boxes, but that's because they are cells inside a wooden box, and the box have regular shapes, typically rectangular or some shape related to a rectangle. But if the cell wall was a solid cell wall, that would simply mean that the plant cells could not communicate with each other. So penetrating through the cell walls are structures called plasmodesmata. And the plasmodesmata allow the cytoplasm of the cells to interact with each other. And it's been determined through very careful electron microscope studies and also molecular studies that the endoplasmic reticulum of plant cells actually connect to each other through the plasmodesmata. So there is a true chemical pathway connection between plant cells, and it allows them to talk to each other and to pass chemicals from one cell to the next, because they don't have circulatory systems like animals do. So this is the way that they can pass chemicals from one cell to the next so that those chemicals can be distributed throughout the body of the plant cell, the plant. And it really does work very, very well because those chemicals can move extremely quickly throughout the body of the plant. In the case of animal cells, there's no cell wall around there, but there is a structural feature around the outside of an animal cell, and it's called an extracellular matrix, or ECM. And this is made up of protein fibers and complex carbohydrates that form an aggregate material that doesn't prevent the plant cell from, the animal cell from swelling, but it does allow the animal cells to stick to each other. It does provide a framework for animal cells to move around, because animal cells will crawl from one location to another during embryological development. They can send signals to each other from the cells as molecules that released into the environment, or the presence of molecules on the surface of cells, receptor proteins and recognition proteins that allow cells to recognize each other and to link up with each other as a consequence, or move away from each other as a consequence. All of this is involved in the extracellular matrix. It's a pretty complicated environment. And we will talk a little bit more about this when we talk about cell membranes in the next chapter, which will be the subject of the next presentation. That's chapter 7. In terms of communicating, we've already talked about the plasmodesmata in plant cells. So you can see the plasmodesmata here in this electron microscope image. And if you look really carefully, here's part of the endoplasmic reticulum of this cell, and you can see that it's coming right up against the plasmodesmata. And if you look carefully on this side, you can see the ER of this cell coming all the way down here. And we know that, in fact, these connect to each other through the plasmodesmata. So there really is a chemical channel for communication directly between the cells via their endoplasmic reticuli. So that is one of the reasons why we know plant cells really do talk to each other very deliberately in a chemical fashion through these plasmodesmata. It's the only form of communication they really have with each other, and it works extremely well. In the case of animal cells, there are 3 different ways that animal cells can connect to each other, and in one case, communicate with each other. So the tight junctions are formed by chains of protein that interweave between the plasma membranes of the 2 adjacent cells. They are really, really tight connections, and these are watertight. And a really good example of these watertight tight junctions are the junctions between skin cells, because we don't want to lose water randomly from our skin. We want it to be a watertight covering so we can regulate how much water is lost. So our skin cells are anchored together by tight junctions, these interwoven chains of proteins that, by weaving backwards and forwards between the 2 groups of cells, or the cells that are being held together, ties them together. It's kind of like stitches holding fabric together in clothing. It weaves backwards and forwards to create a tight connection, so tight that it is literally watertight. Intermediate filaments, or desmosomes, the structural units called desmosomes formed from intermediate filaments, are what anchor cells together. They're not watertight, but they do anchor the cells together. And I'll put up here skin as an example. An example of intermediate or of desmosomes is the links between muscle cells. So these are very, very strong connections, but they are not permanent. They can be broken. And so if you've ever had a muscle tear or a muscle pull, one of the reasons why that hurts so much is because you've broken these connections. You've literally ripped the cells apart. You've ripped these connections, these desmosome connections. And it takes time for this to heal. For number 1, the cells to heal, and number 2, these connections to form up again. And you know, if you tear a muscle sufficiently badly enough, the muscle doesn't work particularly well because you've literally torn the fibers that make the muscle up. So this is a really good example of the desmosome. The final connection is what is called a gap junction, or communicating junction. And this allows chemicals to pass from one animal cell to the next in much the same fashion as chemicals pass from one plant cell to the next via the plasmodesmata. So these are communicating junctions or connections between animal cells that work pretty much the same as plasmodesmata in animals. The big difference, of course, is they don't have to pass them through cell walls. So in fact, in some communicating junctions or gap junctions, most notably those between nerve cells, there is no physical connection between the cells. Rather, the chemicals are released into the space between the nerve cells, called a synapse. The chemicals move across that gap, across the synapse. And then if there are the appropriate receptor proteins on the other side to capture those molecules called neurotransmitters, then the nerve impulse that those molecules represent is passed on into the adjacent nerve cell. So when a nerve pulse comes down a nerve cell and reaches the end of the nerve cell, it triggers the release of these neurochemicals. They pass across the synapse gap. And if there are receptors on the next cell, nerve cell in the daisy chain, the neurotransmitters bind to those receptors and trigger the nerve impulse to move on. And this is how different nerves can pass signals into different sets of nerve nets or nerve complexes and control the passage of those nerve impulses in one direction and another. Sometimes it floods across several sections of the brain. In other cases, it moves in a very particular direction because of the presence and absence of neurotransmitters and the receptors for them in adjacent cells. Again, if you're planning to take our A&P classes because you're going on into the allied health sciences or the nursing program, you're going to learn a whole lot more about these neurotransmitters, the different types of neurotransmitters and how they affect the central nervous system's ability to send nerve impulses backwards and forwards from the brain out into the body and back again to control different parts of the body in numerous different ways. So more to come for those of you who are going to take the AP classes or A&P classes. So one more concept check, a couple more slides and we're done. So protist animal cells and plant cells are all composed of eukaryotic cells. How do cells of animals and plants differ from those found in protists? Give you a few seconds, 30 seconds, and then we'll look at the answer. Okay, so if you were paying attention to the last section, you should have the answer for this. Because remember, plants and animals are multicellular organisms. They're made up of many cells. And they not only need to connect to each other, they need to communicate with each other. In the case of protists, they're all unicellular, single cells. So they don't need to be connected to each other and they don't need to communicate to each other unless they are part of colonial protists. And there are some types of colonial protists, but by and large, the vast majority of protists are unicellular. And so their cells do not need to connect to each other, nor do they need to talk to each other. So they are going to lack the connections that may tie the cells together, and they're going to lack the communicating junctions that might connect them together. Now, that doesn't mean that protist cells don't have the ability to talk to each other, but the way they talk to each other is to release chemicals into the environment and just hope that by random chance, a protist cell of the same species is going to detect the presence of those chemicals. And there are some types of organisms called slime molds. They're somewhat related to animals and to fungi. They're a little bit more closely related to fungi than they are to animal cells or to animals. And they actually live as independent cells while they're feeding, but when it's time to reproduce, they send out chemical signals called pheromones that attracts those cells together so that they link up to form a rather unusual looking aggregate of cells called a migrating slug, because it looks a little bit like a slug. And that is going to give rise to the reproductive structure that's going to allow this slime mold to reproduce. And it's referred to as a cellular slime mold because it exists mostly as independent cells for most of its life, but it only gets together in this slug-like thing during reproduction. And so it kind of blurs the line between what it means to be a unicellular organism and a multicellular organism, which is the reason why they're interesting to biologists, to try and understand a little bit more about being single-celled versus multi-celled. But for most protists, they're unicellular. They don't need to hook up to each other, nor do they need to really talk to each other at length. So they're going to lack those features. So we've looked at all of the components of eukaryotic cells, and we've looked at how all of those different components function. But what we need to remember is that while we might do this, remember this is called the reductionist approach, where we break up a complicated system into smaller units that are more easy to understand, we really only truly understand a cell by looking at how all the various components that we've just discussed come together to interact with each other to create the cell as an integrated unit. And when we start thinking about the cell as an integrated unit, we start to realize that while understanding the various components of a cell gives us some clues as to how a cell does what it does, we really only begin to understand the full impact of all of this integration by looking at the cell as an integrated unit. It's encapsulated in that statement that we saw in chapter 1. The whole is significantly greater than the sum of the parts. And this is very, very true of all eukaryotic cells, whether they be unicellular or multicellular. This happens to be a macrophage. This is one of the white blood cells that is part of our immune system. Its job is to go out and hunt down pathogens. In this case, it's bacteria, to capture those bacteria and kill them by phagocytosing them. That you may remember phagocytosis. We talked about that when we talked about lysosomes. So it captures the bacteria by phagocytosis and brings them into the body of the cell in a vacuole, which is then combined with a lysosome that breaks down the bacteria and kills them off. And this is its job. It is to protect us. So it's using multiple different components that we've talked about: the cytoskeleton, the process of phagocytosis, the lysosomes, which means it's also using the endoplasmic reticulum, and it's using the plasma membrane to carry out this task. So the role of this cell as a macrophage, as a hunter-killer cell, is not seen by looking at any one of those components. It's seen by looking at all of the components integrated together to allow the cell to carry out that function of capturing and killing bacterial cells. The whole, the macrophage cell, is most definitely greater than the simple sum of its parts. And that's something that we always need to remember. So that's it for this presentation. We've now covered the common constituents of eukaryotic cells. We've looked at the basis for some of those, where they came from, what their functions are, what their structures are. We looked briefly at prokaryotic organisms and how they differ from eukaryotic organisms. So we've got a pretty good handle now on what a typical cell, and particularly a typical eukaryotic cell, would look like and what some of its functions are going to be. So when we come back again next week to look at the plasma membrane, we're going to start disassembling the cell and look at individual components of the cells in much more detail, because there are many things that we can learn by looking at them in more detail. And the role of the plasma membrane as a line of demarcation between the inside and the outside of the cell, or the role of the internal membranes as a line of demarcation between the rest of the cell and an organelle, tells us a lot about the functions that can be carried out by a cell or by an organelle. And the role of the membrane as a regulator of what moves into and out of a cell or the compartments within a cell very much influences the chemistry that can take place in the cytoplasm, but also inside the organelles. So that's what we'll address in the next presentation in Chapter 7. What is the structure of a plasma membrane? How does that influence the behavior of the cell? So until next time, come and see me in office hours if you have any concerns, questions, or observations about anything that we have discussed in this presentation or any of the presentations prior to this. Otherwise, I'll see you next time. Bye for now.